Hello, my name is Thierry VandenDriessche. I'm from the Vrije Universiteit Brussels in Belgium. And it's a pleasure for me today to talk about how we can improve the efficacy and safety of gene therapy. I'd like to talk about the current limitations of hemophilia gene therapy. There's been tremendous progress, but we're not quite there yet. And there's a few nuts to be cracked. We need to improve AAV gene therapy further. And I'd like to share with you the current status and future of integrating vector systems. So what are the current limitations of hemophilia gene therapy? Almost 30 years after the initial preclinical proof of concept in mice, gene therapy for hemophilia is becoming a reality. Over the past 2 years, the first gene therapy products for hemophilia A and B have been approved by the European Medicines Agency and the Food and Drug Administration. So this is truly a historic first, and I think this is just the end of the beginning, and we will see more and more of these advances, I believe, in the gene therapy space. We have learned a lot from the, let's say, the first gene therapy clinical trials and the trials that led to the approval of the first gene therapy products. The good news is that we are now able to achieve therapeutic factor VIII levels, 20% at 5 years. So a single treatment that results in 20% factor VIII for the duration of 5 years. I think this really illustrates the potential of gene therapy. Similarly, in case of factor IX gene therapy, even higher levels of factor IX activity were reported, 30 to 40% factor IX at 1 year. And it would appear that these levels are even more stable than in the case of factor VIII. I will come back to that later. The good news is that these therapeutic levels of factor VIII and factor IX result in a significant reduction in annual bleeding rates. There's a discontinuation of prophylaxis in many of the patients that have been treated, and no antibodies were detected to either factor VIII or factor IX, so that's reassuring. Now, obviously, the patients that enrolled in these trials were pre-screened for lack of inhibitory antibodies, so it's not entirely surprising that they didn't develop antibodies after gene therapy. Factor IX seemed to be more stable than factor VIII, and this is really very important for the field at large. But there are some issues that we have to deal with. There's significant interpatient variation, issues with respect to stability, and perhaps most importantly, liver toxicity that has been observed in many of the patients that have been treated. And this liver toxicity is not just restricted to hemophilia gene therapy. It's also been observed after AAV-based gene therapy for the treatment of many other different diseases, such as spinal muscular atrophy, Duchenne muscular dystrophy, and myotubular myopathy. Now, in these trials, even higher doses of the gene therapy vehicle were used. We're talking about 10 to the 14th vector genomes per kilogram. And this didn't only result in liver toxicity, but other serious adverse events as well, related to complement activation, acute kidney injury, hemolysis, thrombocytopenia, GI bleeds, hyperbilirubinemia, and sepsis. Now, obviously, some of these other serious adverse events, even though it was reported in these high-dose trials, hadn't been seen in the hemophilia gene therapy trials. But still, some caution is warranted if we're trying to push the envelope and administer very high levels of vectors to the patients. Now, how can we address and hopefully understand and overcome the remaining challenges? Let's start with the issue of interpatient variation. We don't quite understand why some patients have a very high levels of factor VIII or factor IX and after gene therapy, and other patients do not respond very well. And we can propose a number of hypotheses based on what we know. But we are entering the realm of the known unknowns and the unknown unknowns. So it's becoming a little bit hypothetical at this point. But just based on common sense, we can put forward a number of hypotheses. Every gene therapy trial will require the use of a gene delivery vector in order to bring the therapeutic cargo into the cell. And for the approved products, AAV has been the vector of choice, adeno-associated viral vector. This vector interacts with a cellular receptor and a cellular co-receptor, after which the therapeutic cargo is then trafficked intracellularly into the nucleus, where it is expressed as a messenger RNA, and then translated in the endoplasmic reticulum into factor IX and factor VIII proteins that are eventually secreted by the gene-engineered cells. Now, at each of these different steps, at the level of vector entry, at the level of vector trafficking, or at the level of gene expression, there can be differences between different patients. And these differences, either by themselves or in combination, could account for the difference in factor VIII and factor IX levels that are measured in these patients that were treated by gene therapy. We also cannot exclude a possible role of the immune response that could account for these different outcomes. The next challenge that we have to deal with is the stability of expression. Yes, after gene therapy, there is very prolonged expression of factor VIII and factor IX. There's no doubt about that. But the question is it going to be lifelong? And there is some doubt as to whether we can achieve lifelong expression, because on the basis of some of the trials, there is evidence of a decline in factor VIII expression. We don't quite understand why there is a decline in expression, and a number of hypotheses have been put forward. It could be due to the fact that we are forcing factor VIII expression in a cell that normally doesn't express factor VIII, the hepatocyte. The normal factor VIII producing cells are the endothelial cells, and in particular, the liver sinusoidal endothelial cells. So evidence from preclinical studies suggests that the ectopic expression of factor VIII in cells that normally do not express it, like hepatocytes, triggers a cellular stress response at the level of the endoplasmic reticulum. And perhaps the use of the AAV vector compounds the problem and worsens the stress response. There's also a possibility that an immune reaction contributes to this decline in expression, or some vector rearrangements of vector loss, maybe even differences at the level of vector manufacturing. And last but not least, there's evidence based on biopsies obtained from those patients that were enrolled in these clinical studies that the factor VIII messenger RNA transcription may be shut down. And this gene repression is perhaps the most likely explanation for the decline in expression. Now, how can we explain the liver toxicity that's been observed in many of the patients that have been treated by gene therapy, not just the hemophilia patients, but as I explained earlier, also patients suffering from other diseases that were exposed to very high doses of the gene delivery vehicle? One hypothesis that has been proposed by Cathy Hai and colleagues more than a decade ago is based on the so-called T-cell hypothesis. Once an AAV vector enters a cell, it's chopped into pieces by the proteasome machinery, which acts as a molecular shredder, if you may, resulting in the generation of AAV capsid-derived peptides. And these peptides are then transported across the endoplasmic reticulum membrane through a TAP molecule, and then binds on the MHC class I molecules that are present in the endoplasmic reticulum. These MHC class I molecules that then contain the AAV peptides are then transported to the surface of the transduced hepatocyte. And there they come in contact with the T-cell receptor that recognizes, based on the principle of Zinkernagel's MHC class I restriction concept, the antigenic peptides in association with MHC class I molecules. The T cells, presumably CD8 positive T cells, then are activated and eliminate the gene-engineered cells, resulting in transient expression of factor IX and possibly loss of hepatocytes resulting in transaminitis, so liver toxicity. Now, one can try to control this by tapering doses of corticosteroids that result in a stabilization of factor IX expression. And there's some evidence based on the clinical studies that this is indeed what is happening. However, this is perhaps a little bit too simplistic. And even though T cells may play a role, it is likely that many other immune mechanisms, some of which are unknown, may play a role as well. And why am I saying this? Because there's evidence of transaminitis, so elevations of transaminases without evidence of an AAV-specific T-cell response. So clearly something else must be going on as well. Moreover, corticosteroids are not always effective to curtail transaminitis, and the innate immune response may also play a role. In fact, there's evidence suggesting that the CpG motifs that are present in the vector may activate toll-like receptors which sense this foreign DNA and activate the innate immune system. Even NK cells or interleukin-15 dependent mechanisms may play a role, or at least contribute in part to the liver toxicity and the decline in expression. So further studies are needed to really nail down the molecular mechanisms and the immune mechanisms, or perhaps even non-immune mechanisms that contribute to this liver toxicity. Another issue that we have to deal with is the issue of anti-AAV antibodies. AAV, or the adeno-associated virus, is quite common in the human population. So many of us have naturally occurring anti-AAV antibodies. And the presence of these anti-AAV antibodies may actually preclude gene transfer with AAV-based vectors. So typically, if one is exposed to the wild-type AAV virus, this results in the induction of anti-AAV antibodies. If these patients were to be treated by gene therapy with an AAV-based vector containing either factor VIII or factor IX, they already have pre-existing anti-AAV antibodies in their circulation, and this will eventually result in a further secondary boost of the immune response and also block subsequent gene transfer with these AAV vectors. In fact, this is also the reason why patients that have undergone gene therapy with an AAV vectors cannot be retreated with AAV, because following an initial exposure of the patient with AAV factor VIII or factor IX vectors, this will result in induction of anti-AAV antibodies, and this has been consistently reported in all of the trials. If one were to re-administer this patient a second time with AAV, for example, if these patients were to show declining levels of factor VIII or factor IX, well, this is not going to work because these patients have antibodies against AAV, which will preclude subsequent gene transfer. Again, the subsequent administration of AAV will boost the immune response, resulting in a secondary and more potent anti-AAV immune response. So as long as we don't have any antibodies, that's good news, because there's no blockade then vis-a-vis the vector. The vector can interact with the cellular receptor and co-receptor and deliver its therapeutic cargo into the desired target cell. The hepatocytes can then express factor VIII and factor IX, and as long as there's no immune response against the gene-engineered cells, this will then result in prolonged, if not stable, expression of factor VIII and factor IX. But if you have antibodies against the vector, these vector particles will be neutralized by these antibodies, blocking vector entry and blocking every subsequent step to the extent that factor VIII and factor IX expression cannot occur. So that's the reason why the pre-existing antibodies, we're talking about high titer pre-existing antibodies, are so important and may block the efficacy. Now, with certain vectors and at low doses of certain antibodies, it's still possible to achieve gene transfer. So it's not a black and white phenomenon. It really depends on the avidity of the antibodies, the titer of the antibodies. And I have a few questions as part of our educational objectives that I would like to ask you with respect to this first part of this module. My first question is, what is the most common complication and safety issue in hemophilia gene therapy trials to date? Is it cancer, inhibitory antibodies to factor VIII or factor IX, liver toxicity, or viral replication? The correct answer to this question is liver toxicity. Yes, indeed, that's one of the most common complications. It is coinciding with an elevation in liver transaminases and is likely due to an immune mechanism, even though other mechanisms cannot be excluded. There's no evidence that hemophilia gene therapy would result in cancer. There's no evidence that gene therapy would result in inhibitors to factor VIII or factor IX. None of the patients that were treated show an elevation in anti-factor VIII or factor IX inhibitor titer. And certainly there's no evidence for viral replication because we are using replication deficient AAV vectors. They are unable to replicate.